Biology, Part 208. Number 2, crossing over. Crossing over or chiasma formation takes place following synapses. In this process, homologs, chromatides, break and rejoin at precisely corresponding points so that genetic material is exchanged between them in the figure. Crossing over is a random event leading to the genetic recombination, that is, it generates a new combination of genes. The crossover, also called as the chiasmata, singular chiasma, can occur anywhere along the length of the chromosome. Typically 2 or 3 chiasmata are found on each pair of the chromosomes in production of the human gametes. The gametes which contain new arrangements of the genes are called the recombinant types. Prophase 1 normally lasts for several days. In its later stages, the chromosomes become fully condensed and undergo the desynapsis, remaining linked only where the chiasmata have been formed. The subsequent stage of meiosis, one separate the members of each homologous pairs. Note carefully that it is entire chromosomes, each consisting of 2 daughter chromatides, which are segregated into the daughter cells of the first division. The second division is called the meiosis II, is very similar to mitosis and serves merely to separate the chromatides, resulting in the production of 4 haploid gamete cells. Evolution of meiosis. In the context of evolutionary development, the mechanism of the meiosis represent a series of elegant adaptations to the problems of rearranging and transmitting genetic information in sexual reproduction. The first eukaryotic cells reproduced asexually and were undoubtedly haploid, having only one gene for each of their characteristics. In such organisms, a change or mutation in any particular gene will directly affect survival. Unfavorable mutations may be lethal, while favorable ones confer an immediate advantage. Population of the organisms with different favorable characteristics compete with one another, but except by repeated mutation, there is no way of combining favorable characteristics in the same individual. Evolution is slower because mutation is a random process and beneficial mutations are rare. Fusion between the haploid cells may have given rise to new types of organisms, each with a pair of genetically different nuclei. These individuals would combine the favorable characteristics of the parent lines and would have an obvious selective advantage. Furthermore, because they now have 2 genes for each of their characteristics, the new organisms are protected against the most damaging effects of additional mutations. Thus, one copy of each gene is free to mutate without the loss of the viability and may ultimately come to serve a new and useful function. Similar processes still occur in fungi, many of which exist at eukaryotic individuals, Unit 46. Initially, the two nuclei in each cell might have divided separately, but in the later evolution, nuclei fused so that the two complete sets of the chromosomes became attached to the same spindle during the mitosis, forming a true diploid organism.